You leave it out for about 20 hours, things fly towards it, they get sucked into a fan, you come back the next day and you have a net full of bugs. Okay, an entomologist takes this back to a lab, knocks everything down in the net, and then under a dissecting microscope sorts them out, and then some of them go for testing to see whether they're infected with something. So that is the state-of-the-art method today for surveilling any mosquito-borne disease in the environment. It's slow. Even if you have the nets and the fans, many places don't have the expertise to process the samples that they retrieve. So the first thing we needed to do was to build a smarter trap. And that's what you're seeing here. This trap is essentially a robotic field biologist. It has 64 of these smart cells on it that work like little Venus flytraps. When something flies into one of these cells in real time, it tries to tell what sort of insect it is, and if it's something that it finds interesting, it closes a little door and captures it, giving us a sample isolated in each of these little chambers. Now, in addition, because every sample is separated, we get a whole bunch of additional data about this sample. We know the exact moment it arrived. We know the ambient light levels, humidity, temperature, barometric pressure, everything, all the conditions that were right for that sample arriving to the trap at that particular moment in time. This is data about basic vectors. When I say vectors, I mean the sort of insects that carry disease that has never been seen before. Again, our initial motivation was, impetus for this was a response to the Ebola epidemic. This is an early test of something similar to this device with our collaborators at University of California, Riverside on Aedes aegypti mosquitoes, which are the ones that carry Zika and dengue. You can see the mosquitoes floating around the device here. Let me just summarize how this works. Each of these smart cells has an array of infrared LEDs on one side. They emit an infrared plane that's detected on the other side. When something flies through, it effectively casts a shadow. And because its wings are flapping, the amount of light that's blocked as its wings flap changes as a function of time. And that turns out to be a fairly representative signature that allows you to tell things apart. So if you just look right here, you can see that little Aedes aegypti flying in and getting caught. If you put your fingers in that, nothing happens because it tells it apart. Okay, so we were doing that, and then it became clear that actually the next candidate emerging infectious disease we would be worrying about would be Zika. One of the largest health organizations in the US, Harris County Public Health and Environmental Services, approached us. They're the health organization that monitors mosquitoes in Houston. And they asked, could you deploy this system in Houston as we're worrying about Zika possibly emerging there? And so we went from that prototype you saw in the lab to the one that you see here, which is a fully ruggedized field deployable version. Ran 87 experiments over during last summer, during which we collected over 20 gigabytes of data about the mosquitoes in that area, detecting over 22,000 mosquito events. What can we learn from that? What we can learn from that is, using machine learning in the cloud, is we can build very predictive models that explain what factors drive the presence or absence of important disease vectors in the environment. Now, I don't have time to walk through every one of these graphs, but let me just try to briefly summarize. The greenish line is a genus of mosquitoes called Aedes. Aedes aegypti and Aedes albopictus are the ones that carry Zika. The red is a genus of mosquito called Chalice. They're the ones that carry things like West Nile. And what you're seeing here is how we can use machine learning to explain at points in time and different environmental factors when these will show up in the environment. So for instance, we can know very precisely that if the conditions on a particular day look like this, and you're standing in the place in a point in time, you have a very high probability of encountering a mosquito that might carry Zika, or you have a very low probability. So you can imagine the utility of this data both from the public's point of view and from the public health point of view that needs to make a decision, where do we go and when do we stop a mosquito population that might be a vector for a disease? So just a really brief summary of what we saw from that initial experiment is we can get lots of, well, first we get lots of mosquito encounters, which might not be surprising in a place like Houston, but we have a very high predictive capability to in real time tell them apart. So with over 90% accuracy, we can tell apart in real time the kind of mosquitoes encountering that device in Houston. And that's probably approaching what a human would do. The samples that we get back are very high quality because there's no sort of fans blowing these things, so it's very easy to produce training sets. And we get a lot of abiotic data to build models of their behavior. Okay, but I started with the idea of using Mosquito as a device, and what we sort of learned along this path is that just data about mosquitoes is already incredibly useful, but we still wanted to explore the idea of using them as a device. So we get them back and we turn them to data. And by turning them into data, it means we make mosquito juice. That's a vial of mosquito juice there, and it goes into a gene sequencer, and then what comes out is digital data that represents a mixture of genetic material that was co-located with that mosquito. So if it did something, it has a mixture of its own genomes plus the genomes of whatever it's consumed, plus the bacteria in its gut. It's a big, complicated mixture. And what we want to do is sort out that mixture to know if there's anything interesting in that mosquito. This turns out to be a very hard problem, and this is the way we think about it. You give us juice, we actually don't care where it comes from. In this case, we got it from mosquitoes. And we want an explanation of the recipe that made up this genetic material, this mixture of genetic material.